Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Eugenia, and I'll be your host. It's our pleasure to have Solvias with us today, who will be presenting this webinar titled Antibody-Drug Conjugates, Overcoming Analytical Challenges in Next-Gen Cancer Therapies. Let me introduce our esteemed guest speaker for today, Christoph Lederer, PhD, Head of Operations Separation Analyzer at Solvias. Now I'll provide some housekeeping information before we start. First of all, I would like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you have received by email to rejoin the session. During today's event, there will be plenty of time to ask questions to our speaker. You can do that via the questions widget. Just type your question in this box and click submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. We'd also like to invite you to fill out a short survey at the end of the webinar. If you require any assistance, please use the Help widget, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Christoph. Over to you.
Thank you, Evgenia. So, hello, my name is Christoph Lederer. I'm an operational leader at a CRO called Solvias, which is located in beautiful Switzerland in the close proximity to Basel. Head of operations in a CRO means I'm not producing drugs. My team and myself are taking care of analytical support of our customers. So today we want to talk a little about antibody-drug conjugates and the analytical challenges we, especially as a CRO, but also the whole industry is facing when testing them for CMC or characterization purposes. So during the next minutes, and I hope I will sticking to 25 to some for question and answers. I will a little bit go into detail why ADCs are at all interesting for the pharmaceutical industry and actually for investors especially. I will go a little bit into more beginners topics like ADC structure because the audience might be quite broad today. So the whole talk is held to an overview to give an overview and will not go into the detail of a single aspect of an analytical challenge. This could be done in a further more deep dive talk because more or less about every challenge you can do such a webinar. So we will start going a little bit into the details of an ADC structure. What does an ADC make an ADC? Then we will talk about challenges and advances in ADC technology. So what makes ADC as an API complicated to deal with, or what are the challenges that one might need to overcome or think about? Then what are the most recent advances and trends we see in the ADC field? CMC considerations, as this is what Solvias, my employer and myself are dealing with most of the time. In the end of the talk, I will talk a little bit about Solvias ADC portfolio. I will introduce to you our new toxin handling facility because toxicity is something which is very prominent when dealing with the analytical support of ADCs. And I will take one slide to just introduce to you who we as Solvias are. So, why are ADCs interesting? First, ADCs are mainly surfaced 2 decades ago or even a little bit earlier because the oncology market was dominated by very harsh side effect products like usual anti-cancer treatments that are quite inspecific and could not be that potent. So with the introduction of antibodies as a usual compound, more or less is state of the art, is more or less baseline biologics market is antibodies we can test back and forth with very platform methods. So with this compound having controlled, it was clear that we want to try as an industry to use the specificity of antibodies with high potency drugs, so that we have this magic bullet that can really direct it, affect the tumor where it is. In nowadays, it's not only oncology where ADCs are used, there's also like inflammatory diseases and things like that, but it's still mainly an oncology drug, and today I will mainly focus on oncology treatments. So it is a super interesting new modality. It's still considered a new modality because the baseline effect, we understand how it works, very precisely understand how it works. We can very much control it analytically. And that's a big difference, for example, to the cell and gene therapy market, which is another big coming up modality. Today we have ADCs approved worldwide. It has been 15, one had to be taken back. And we have more than 100, I would say even more than 200 molecules in various clinical stages of development. Why is the ADC market so interesting for investors? And at the moment, where investor money is very, very short and investors are not so keen in investing into high risk modalities, of course, the of 25% and the fantasy of a 30 billion market in 2008, which some of the projections show, is very interesting. But what is key for investors is the success rate in clinical phase trials. So it is more or less triple as high as for other oncology products in phases 1 and phases 2. In phase 3, this is more similar because if you have once gone through phase 2 and the efficacy of your product is proven, phase 3 has a way higher success rate than earlier stages. But especially in phase 1 and phase 3, the success rate of ADC products is dramatically higher than for other oncology products. So this makes it very interesting for any investor to invest into the market, and it makes it interesting for upcoming biotech to do the science on new molecules, because they have a highly likelihood, if they're successful to the first 2 phases, to be bought, and they have a return on their investment. So what makes an ADC an ADC? So first you have the antibody backbone. Very many of these antibody backbones are drugs themselves. But what makes it then an ADC is the drug linker, or a linker drug construct, which is a small molecule usually. And therefore, as I'm a large molecule expert, I will mostly talk about the full ADC analytics or the antibody backbone analytics. The chemistry analytics done on linker or drug linker is mainly done by drug linker producers. So very few of them is outsourced. So therefore, it's not a big topic for Solvias. However, at the end of the focus also for our customers is usually on the whole molecule, and not so much on the small molecules compound. What are general challenges in the ADC technologies that we're facing? So this is this slide, and the next slide has 2 main sides. On the left side, you will see challenges that more or less affect safety. And on the right side, you see challenges that affect efficacy. So one of the biggest ADC challenges that's coming with the high potency that we have on the payloads is pharmacokinetics. So where the drug goes in the body and where it stays is crucial for an ADC product because the drug is so potent. We cannot have the antibody unload its drug at the wrong places. So the ADC performance is highly dependent on the antibody type, depending on the backbone and the binding site. So any variability, be it intentional through mutations or stability changes, is absolutely necessary to control, because if the drug goes to the wrong position, this can have severe side effects. And the same is if the linker is not stable enough, for example, and we lose our drug at stages or in areas of the body where we don't want to lose it. So off-target toxicity is usually the case when we have a premature payload release, and this, again, severe side effects are the cause of that. On the efficacy point of view, it is very important that at the right side, the load is also released. So also a tool-tight linker can be absolutely not beneficial. It's then mostly not a safety problem, but an efficacy problem. And this is especially true for solid tumors, where with the bigger constructs, only a small portion of the ADC will penetrate the tumor anyway. And of course, as for every drug, cancer cells can develop resistances, like upregulating efflux pumps or altering gene expression. Luckily, over the last years, we have made a lot of progress in the ADC technologies that we can use today for drug development. So site-specific conjugation helps a lot controlling the drug-antibody ratio. So if you have a non-specific conjugation, your drug-antibody ratio can go through the roof or be too low, which again has effects on either efficacy or safety. So we have a lot of engineered cysteine residues, cysteine rebridging, or amino terminal engineered serines that are applied all over the field today. Something that is an analytical issue or an analytical challenge, hydrophilic linkers helped a lot against the hydrophobic aggregation forcing linkers that were used in the early development stages of ADCs. However, they are then not so easily distinguishable for DAR ratio analysis, but they, of course, help with pharmacokinetics and aggregation. And a lot of the new generation payloads, they no longer are pure cell poisons, but they can affect immunocheckpoints, can be inhibitors, RNA inhibitors, PROTACs, or we even have dual payloads if the effect is not big enough with a single payload, or to overcome drug resistance. A big point, especially for the solid tumors that I mentioned shortly before, are lower molecular weight target molecules. So this means the target function of an antibody is then replaced by an antibody fragment, even a peptide, or just a FAP. This helps, the smaller molecule size helps actually by tumor penetration and can also enhance site-specific payload delivery. Another point of CMC considerations is that we need to understand that an antibody-drug conjugate is, just from the sheer analytical burden on a submission, more complex than a usual antibody project. So you have a drug linker synthesis. You need to control that your drug linker is to your liking and to your expectation. You have a point of control on that. Again, that's usually done by the producers of these small molecule compounds. But you also have, of course, on the map itself a production cycle. Everything which you needed to do also for a pure map project, you would also need to do here. So you have a production, you have an upstream and a downstream process, and you also would have your specification and testing. The conjugation process is then followed by a drug substance release, and then it can be further formulated. Drug product can be produced, and also devices can be used, like pens, EpiPens, or things like that. So then you have a usual drug substance and a drug product release. The naming conventions for all these stages is not finalized. So some use the naked antibody or the antibody scaffold and call it drug substance, and the conjugated antibody call it drug product. So everything is existing in the market, but usually that kind of naming is used. So subparts of your ADC product, when we talk about the targeting molecule, the antibody, it's mainly about integrity. So the antibody is usually very well established and very well already tested. So sometimes also historical data can be used for the testing of this antibody, and it's all about the integrity of the molecule. The linker is usually tested for stability, and the payload is, of course, tested for potency. And ADC-specific CQAs who are really important are the drug-antibody ratio, which needs to be monitored about along the whole life cycle of an ADC product, but also CBPA. And with the payload, CBPA tests usually go into the direction of killing assays. Also, the usual ADCC and CDC antibody like CBPAs can still be also important for the potency of an ADC product. So if one looks more on the CQA level, and I will now not go so much into in detail for the drug linker intermediate, but if you look on the CQAs for the MAP intermediate or the ADC drug substrate and drug product, for the MAP intermediate, it's the whole testing panel for an antibody. Appearance, of course, all the pharmaceutical or the pharmacopoeia methods are applied. Purity, impurity, like size and charge, host cell proteins, host cell DNA, residual protein A, depending on the products, on the production process of the MAP. Biological activity and primary structure are of concern. For the ADC drug substance, appearance is also important, but then other CQAs come into play, like the drug-antibody ratio, or free drug substance, free drug analysis on the impurity portion take a more important role. Beside all the testing panel, also again you would apply on a regular antibody. For drug product after formulation, the usual types: description, residual moisture, sterility, CCIT. Every antibody product is still true for an ADC product. The potency depends on the mode of action of the ADC product. So when one looks to certain CQAs and what kind of analytical methods or technologies can be used to elucidate them, size variance always a topic, especially with hydrophobic linkers in ADC products. So these are usually done by SEC, CSDS, AUC, but of course aggregation can also be used by SEC mals and AF4 mals. So whatever the molecule needs, the usual aggregation technologies are also in place here. Charge variants are usually done by ion exchange chromatography like KEX and AEX, CIF, or the more newer version with the Maurice system, ICIF, and of course CZE. The unconjugated map is also tested by different analytics. I will not go into detail now. The free drug analysis is usually done by RP HPLC, but can also be done by 2D LCMS. And the drug antibody ratio can be done by various different methods, and I will go into detail of some of them in the next slides. So talking about ADC, the drug antibody ratio is most likely the one analytical approach or the one CQA that is the most important in such a development exercise. And there are different analytical modes that can be used, from very simple UV-Vis approaches, where you just need to make sure that you have a wavelength maximum that you can utilize for your analysis. Then you have a very easy way of defining the drug antibody ratio just by the higher absorptions you have with higher drug antibody ratios. You can also, and that's the most common analytical approach for drug antibody ratio, you can use HIC approaches where you don't need a sample preparation, and you also can detect unconjugated maps. So you have a DAR0 variant within this analytical technology. It is a little bit, the downside is that you have a complex workflow to ID the peaks, and you have a high salt mobile phase, which does not make it that easily connectable with MS, which is the highest advantage for the reversed phase approach. You have a high resolution for the reversed phase approach, and you can also very easily apply it with MS to have a one-to-one peak characterization done directly via that. But you have no DRA distribution on the intact level here, and the data processing can be somewhat complicated. Also, especially here in Solvias, we have an excellent MS department that can solve this problem for you. Problem with reverse phase is hydrophilic payloads, because then you have only small changes in hydrophobicity, or even dual payload ADCs, because the hydrophobicity mainly does not discriminate between the different payloads. Another very important CMC test, which is crucial for the quality of the product, is the free drug analysis. So free drug can come into your samples in two different versions. It can be a process impurity of your conjugation process, and it can be a stability impurity because of linker breakage during storage. What we usually do when we test for free drug within our analytical method is we separate the free drug small molecule from the large molecule on our columns, for example, and then we get a known peak, or if you, for example, use an RP reverse phase for that, we can even discriminate between several small molecule free drug variants, like the free drug or without the linker or with the linker, or even MAC adduct or similar can then be discriminated already on that phases. Again, this can then also be coupled with MS. Also, it is very important to state that when we deal with higher amounts of free drug, the high potency of this molecule makes toxicity, also to conserve your employees from being poisoned by these high potent drugs, makes MS sometimes a little bit difficult for these kind of applications. But usually it's not necessary. Charge variants are done by the usual suspects. So you have the ion exchange chromatography, the isoelectric focusing, and very rarely used but possible in special cases where CIF or IEX would not lead to a good resolution. You can then use capillary zone electrophoresis. All of them have upsides and downsides, while the ion exchange chromatography does not need a special sample preparation and is very easily enrichable. So if like fractionation experiments are something that needs to be done after that, the high resolution of isoelectric focusing makes it usually the way to go also for drug antibody, for ADC products. What you always have for isoelectric focusing is a lot-to-lot for your amphelite sensibility, but this is then just a critical reagent one needs to, and usually it does not create bigger life cycle problems. The things that would impact you in an ADC molecule with your charge profile are conjugation events like lysine conjugation or site-specific cysteine conjugations, or even hinge cysteine conjugations. All of them have a different influence on your charge pattern, and therefore charge can be the more challenging CQA to test than, for example, size. And with that, I want to switch over to what we as a CRO can offer to our customers in regard to ADCs in special or in biologics in the broader range. So Solvias is a one-stop shop characterization and CMC testing CRO. So we can support our customers from early phase initial force degradation studies, early phase method development, first in human phase 1 clinical trials, method setup, qualification, or let's say phase appropriate validation, full validation, and we can be your partner until the very end of a commercialization. We also test today, we have ADC products where we test commercial release for some of our methods. We have also partners which we brought successfully through phase 1 and 2. So we are your partner for the whole way. Doesn't matter on which stage you want to join us. So we have a full ADC analytical testing panel available for ADCs. We have a full primary structure package. We can do heterogeneity and PTMs with different methods. The PTMs can be either the usual amidation or oxidation, or also carbohydrate structures and conjugation conformation. Whatever is needed in your special product, we can help you with our analysis. We have also a full higher order structure panel. Mentioned is here CD, DSC, and HDX, but we also have, of course, all the other necessary or not necessary technologies like SECMALS and AF4MALS. Also for charge, we can offer each modality or each technology which is the best for your product. The purity can be done by size or by hydrophobicity. And we also can offer different size heterogeneity or functionality testing already during the characterization portion. For the release and stability testing, we can offer you full panel testing There is no test that we don't have here in our site in Kaiser-Augst or in our partner site in Homburg, which is 30 minutes away. So when you send us your product, when you have a project with us, the whole analytical package is tested out of one hand and mainly out of one site. So not only can we offer the API testing, but we can also offer the process impurities, excipients, or even formulation excipient testing, whatever is needed. We are very proud of is when we're talking about ADCs and we talk about toxicity. Solvias is now the second provider in Switzerland who has a state-of-the-art level 4 toxicity lab, which gives a shell-in-a-shell concept for even solid form free toxins or free payloads. In such a situation, the solid form payload would arrive at Solvias. We would introduce it in a specified container into our class 3 biosafety cabinet, where we would resolve the solid form free toxin, and then the sample preparation would take place in this class 3 biosafety cabinet. In the sample preparations are then allowed to come out of this class 3 biosafety cabinet, but need to say in the sealed off laboratory, and all the testing would then take place in this second shell. The full ADC takes other precautions, and in our case we call this then TOX 3 environment, which is also a two-level concept. For our TOX 3, we would use compounds down to an OEL of 50 nanogram per cubic meters. And in this new facility, we can also handle compounds down to a toxicity level OEL of 5 nanogram per cubic meter. So Solvias is a world-class CRO with sites all over the planet now. We have sites in Europe with our main site in Kaiser-Augst and Homburg, which is all in the Basel pharmaceutical hub. We have a site also in Utrecht. We have now also a very new footprint in the US, in the Research Triangle in North Carolina, in Raleigh, where we more or less want to mirror the existing portfolio we also have in Europe in Kaiser-Augst. There is also a small E&L site in Canton, a little bit above that now. We have roughly 175 PhD level scientists, and in total over 23,000 square meters of lab capacities. And with that, I thank you for your attention, and I'm happy to answer your questions in the next few minutes.
Yeah, thank you very much for this presentation. Just a reminder to the audience to ask questions. You can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they will be answered at a later date. And it looks like we've got several questions already, and here's the first one. Considering all CQAs of an ADC CMC package, which of these is the analytical most challenging, and how does Solvias address these challenges?
Yes, so the most challenging CQA to test is for sure the free toxin testing, because it affects not only also has a toxicity a level that one needs to consider. And we at Solvias have invested into our ADC portfolio by investing into this top-notch toxicity environment.
Yeah, thank you very much for that response. And we have another question from the audience. Considering toxicity, the free toxin handling is done in a special containment in Solvias. Are there also toxicity consideration made for the complete ADC?
Yes. So the complete ADC is always also evaluated by our Health and Security and Environment Department, Health, Safety and Environment Department. And if it is near the 50 nanogram per cubic meter OEL, we also apply a two-level approach. So we have special tox-free environments where we would then take the solid form of the ADC, resolve it in this tox-free environment, and then we can bring the resolved in our usual lab environments.
Yeah, thank you very much. And we have another question from the audience. Is there a more unusual analytical technique you see that could be especially beneficial for ADCs compared to standard biologics?
So, quite rarely used MS approach, which could be very interesting for ADCs, is HDX, which is hydrogen deuterium exchange mass spectrometry. This is a technology where a PEPMAP is done after soaking the product in deuterium for a defined time frame. With that, you can find out, for example, if you compare the naked antibody with the conjugated antibody, what kind of areas of your protein are affected in accessibility by the conjugation process.
Yeah, thank you very much. And we have another question. What is it for you personally that makes you excited about ADCs as a life-changing drug modality for many patients?
So we at the moment have two big trends in the pharmaceutical industry. The one trend that we have is a cell and gene therapy approach that is something which is very, very hyped, but we do not understand that well. On the other side, we have the ADCs, which makes me very excited because they are highly potent. We are still in the beginning of modifying the ADC technologies and still bringing new and interesting ways of addressing special kind of sicknesses and illnesses. And as we understand the backbones and also how we need to test and analyze and control the production and also the administration of such kind of molecules, I'm very excited about that personally, because I think that this will be the future of the cancer treatments.
Yeah, thank you very much. And I believe we still have time for one more question today. So please keep sending us questions. And our next question is this one. Your recommendations and most accepted general and specific analysis over the ADC production steps, employee and site protection.
Yes, so this is a very big question. I will try to keep myself restricted. So when one looks on the backbone of the map, the usual suspects for a testing panel at least for the release specification setting, are there. So you would have your SEC, you would have your CIEF, you would have a PEPMAP ID in any way, shape, or form. And you, of course, need to test for all, on a drug substance level, for all the process impurities, and you need to do your microbiology testing and also your appearance testing. So these are would be a usual set of analyses. For an ADC, what comes on top is, of course, then the drug-antibody ratio, which is usually at the moment done with HIC, but preferably done by RP. And of course, the free toxin testing, which at the moment is usually done by reverse phase HPLC approaches. And yes, the site protection is really something that needs to be considered when dealing with ADC products, because it's not usually, sometimes producers of such ADCs are not, they do not consider that the CRO they send the samples to do not have the same safety precautions everywhere as they might have in their production environment. So this is always a topic that needs to be at the beginning of a project very much considered. But we can say at Solvias that we are prepared now after the investment into this state-of-the-art toxicological level 4 environment, that we can handle all of that. And the employee protection is key. So the employees that are, for example, testing in such a Tox 4 environment, they are specially trained and highly educated in the danger of the samples they are dealing with, and at least it's Solvias, they also get a special benefit by working in that environment, because there is a full, it's a full body safety protection with goggles and everything. So it's not that nice to work in that environment. So therefore, there is some benefits on that.
Yeah, thank you very much. And just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. And I would like to thank Christoph for this presentation and Solvias for sponsoring this session. To that, indeed, you will receive an email with instructions on how to access the on-demand version of this webinar. Alternatively, you can access it through our website, which is pmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.
Thank you all. Have a great day.